There is a difference between a dead plant and a dying one, and most people on the internet cannot tell them apart. A plant with fully rotted, brown, structurally collapsed roots is biologically dead. No fertilizer on Earth will reverse cellular decomposition, but a plant that is yellowing, drooping, or refusing to grow, that plant is not dead. Its roots are likely locked out of the nutrients already sitting in the soil, unable to absorb them because of pH imbalance, microbial collapse, or physical damage from overwatering. Those are two completely different problems, and they require two completely different responses. If you have spent money on blue synthetic fertilizer, dumped it on a stressed plant, watched it get worse, and felt confused, this video will explain exactly why that happened and what to do instead. Today we are going through 21 fast-acting, scientifically grounded, zero-cost fertilizer methods drawn from verifiable horticultural science. For every single one, we will explain the exact biological mechanism, what compound it delivers, how the plant absorbs it, and where most people go wrong. We are also correcting the most dangerous myths currently circulating online that are actively destroying gardens, starting with the most important correction of all. Synthetic nitrogen fertilizers applied to a stressed, root-damaged plant do not feed it. They increase salt concentration in the soil, pull water out of the root tips through osmotic pressure, and accelerate the exact dehydration, already killing the plant. If your plant looks like it is burning from the inside out after you fertilized it, that is salt burn. This video is the correction. We begin with liquid methods because liquid delivery is faster than solid. Plants absorb dissolved nutrients through root hair cells by diffusion and active transport. They also absorb certain compounds directly through the stomata, the microscopic pores on the underside of leaves. Foliar absorption can be up to 10 times faster than root uptake when root systems are compromised. This is established foliar feeding science that has been used in commercial horticulture since the 1950s. Every liquid on this list works because it delivers something specific. Let us begin. Number 1. Cooled, unsweetened green tea. Green tea contains tannic acid, specifically gallatannins and thiogallin. When poured around acid-loving plants like blueberries, camellias, or azaleas, tannic acid temporarily lowers soil pH. This matters because iron becomes chemically unavailable to plant roots above a soil pH of roughly 6.5. By lowering pH slightly, the iron already present in the soil is converted from ferric to ferrous form, the only form roots can absorb. This directly resolves the iron deficiency chlorosis pattern you see in acid-loving plants, yellow leaves with dark green veins. Use cooled, plain tea with no sugar and no milk, 1 to 2 cups per medium pot, once per week. Number 2. Water drained from a freshwater fish tank. Fish waste contains ammonia, which is broken down by nitrosomonas and nitrobacter bacteria already living in aquarium gravel into nitrite, then nitrate, a direct, plant-available form of nitrogen. This water also carries those beneficial nitrifying bacteria into your soil, helping rebuild a collapsed soil food web in sterile, overworked potting mixes. The key word is freshwater. Saltwater aquarium water contains sodium and chloride ions that are toxic to terrestrial plant roots and will not wash out easily. Use it undiluted, directly in place of a regular watering. Number 3. Heavily diluted liquid black coffee. Coffee contains nitrogen in the form of proteins and amino acids, and it carries a mild acidic pH between 4.5 and 6, depending on brew strength, which helps acid-loving plants access locked minerals. However, and this is the correction that matters, do not dump wet, used coffee grounds directly onto the surface of struggling potted plants. Used coffee grounds clump into a hydrophobic mat. Water beads off the surface instead of penetrating, and the dense, moist organic layer creates an ideal environment for fungal mold to colonize the root zone. Instead, steep 1 tablespoon of grounds in 1 liter of water overnight, strain it completely, dilute with 4 additional parts water, and apply that liquid to the soil. Number 4. Epsom salt spray. Epsom salt is magnesium sulfate, the exact compound represented as MgSO4. Magnesium is the central atom in every chlorophyll molecule. Without it, the plant physically cannot synthesize the green pigment that captures light for photosynthesis. The visible symptom is interveinal chlorosis. The tissue between leaf veins turns yellow, while the veins themselves remain green. Dissolve 1 tablespoon of Epsom salt in 1 gallon of clean water. Spray it directly onto both sides of the leaves, particularly the undersides where stomata are more concentrated, because you are bypassing the root system entirely and delivering magnesium directly to the leaf tissue. You can see visible greening begin within 48 to 72 hours on plants with mild deficiency. Do not apply more than once every 2 weeks. Excess magnesium competes with calcium uptake in the roots. Number 5. Fermented rice water. When you wash raw rice, the cloudy water that results contains soluble starches and simple carbohydrates. Left at room temperature for 24 to 48 hours, naturally occurring lactobacillus bacteria begin fermenting those starches, producing lactic acid and a range of bioactive compounds, including beneficial enzymes. When applied to soil, these compounds selectively feed mycorrhizal fungi, the underground fungal network that extends a plant's effective root surface area by up to several 100%. Healthy mycorrhizae dramatically increase the plant's ability to absorb phosphorus, zinc, and copper that would otherwise remain chemically locked. Use it within 3 days of fermentation beginning. If it smells like vinegar, it has gone too far. Dilute it further or discard and restart. Number 6. Willow bark water. This is not folklore. Willow species contain 2 well-documented compounds relevant to plant propagation: indole 3-butyric acid, known as IBA, which is the same active ingredient in commercial rooting powder, and salicylic acid. IBA stimulates meristematic cell division at root tips and cut stem surfaces, directly triggering the formation of adventitious roots. For a plant suffering from root rot, where much of the original root structure has been lost, this can mean the difference between recovery and collapse. Snap young, green willow twigs into segments and steep them in room temperature water for 24 to 48 hours. Use this water when transplanting damaged plants or water struggling plants with it once per week during recovery. Number 7. Aspirin water. This one surprises people, but the mechanism is well established in plant pathology. Aspirin is acetylsalicylic acid, which the plant converts to salicylic acid, the same signaling compound plants naturally produce when under pathogen attack. Salicylic acid triggers a state called systemic acquired resistance, or SAR. The plant upregulates its own immune pathways, increases production of pathogenesis-related proteins, and becomes measurably more resistant to fungal infection for several weeks. Dissolve 1 uncoated regular aspirin tablet in 1 gallon of water. Do not exceed this concentration. Higher doses of salicylate are phytotoxic and will suppress growth instead of stimulating it. Apply as a soil drench or foliar spray once every 3 weeks. Number 8. Heavily diluted milk spray. This is one of the most well-documented home remedies in horticultural science, with multiple published trials showing effectiveness against powdery mildew comparable to synthetic fungicide treatments at low concentrations. Milk proteins, particularly lactoferrin and immunoglobulins, combined with exposure to ultraviolet light from the sun, create a mild antiseptic effect on the leaf surface that disrupts the spore germination of powdery mildew fungus. The dilution ratio is critical, 1 part milk to 9 parts water. Applied at higher concentrations, milk proteins decompose rapidly on the leaf surface and create an ideal environment for bacterial growth and leaf burn. Spray the upper and lower leaf surfaces in morning light so it dries fully before evening. Number 9. Unsalted, fully cooled starch water from boiling pasta or potatoes. When you boil starchy vegetables, soluble carbohydrates and simple sugars leach into the water. These sugars function as an immediate energy source for the microbial community living in your soil, bacteria, fungi, protozoa, and nematodes that collectively make up the soil food web. A collapsed or sterile potting mix has very little microbial activity, which means it also has very little nutrient cycling. Feeding the microbes is not a secondary strategy here. It is the strategy because microbes are what convert raw organic matter and locked soil minerals into plant-available form. The only absolute rule: never use pasta water that was salted. Sodium ions at concentration are lethal to soil biology and accumulate with each watering. Those 9 liquid methods each work through completely different mechanisms, pH adjustment, direct mineral delivery, foliar absorption, microbial inoculation, hormonal signaling, and immune activation. But most people watching this have heard about a completely different category, the solid scraps. Eggshells, banana peels, coffee grounds, wood ash. And this is exactly where things go wrong. Here is the principle that governs the next section. Raw solids do not feed plants. Microbes feed plants. The job of the gardener is to get nutrients into a form that the soil food web can process and that roots can physically absorb in meaning dissolved, ionic, water-soluble form. A whole banana peel sitting next to a root does not release potassium into the soil. It sits there, attracting fruit flies, blocking oxygen exchange, and slowly molding. The fertilizers in this section all work, but only when prepared correctly. Skip the preparation step, and you are either wasting time or actively damaging your soil. Number 10. Water-soluble calcium from dissolved eggshells. Crushed eggshells are primarily calcium carbonate. In dry, solid form, calcium carbonate is insoluble. It takes years of microbial decomposition and acid weathering to release meaningful calcium into the soil. If your tomatoes or peppers are showing blossom end rot right now, waiting 2 years is not an option. The solution is to dissolve crushed eggshells in plain white vinegar overnight. The acid-base reaction converts calcium carbonate to calcium acetate and releases carbon dioxide gas. You will see vigorous bubbling. When the bubbling completely stops, all accessible calcium has been converted. Strain the liquid and dilute it with 10 parts water before applying. Calcium acetate is water-soluble and immediately available to roots. This is the only preparation method that makes eggshells useful in less than a growing season. Number 11. Strained wood ash tea. Wood ash is genuinely useful. It is rich in calcium carbonate, potassium carbonate, and trace minerals. The problem is that raw ash is highly alkaline, with a pH that can reach 10 to 12. Dumping it directly on the soil around a struggling plant can produce an immediate, severe pH spike that locks out every nutrient simultaneously. Always steep wood ash in water, roughly 1 cup per 5 gallons, for 24 hours, then strain it completely. Use only in garden beds where you know the soil is acidic and the plants tolerate alkalinity. Never apply to blueberries, rhododendrons, potatoes, or raspberries, and do not use it more than once per season per bed. Number 12. Fermented deeproot weed tea. Plants like dandelion, dock, comfrey, and nettles have taproots that extend several feet into the soil, accessing mineral layers that shallow-rooted crops never reach. They physically pull up calcium, magnesium, iron, and trace minerals and concentrate them in their above-ground tissue. When you steep these plants in water for 1 to 2 weeks in a covered container, the tissue breaks down and releases those minerals into the water. The resulting liquid is a broad-spectrum trace mineral supplement that commercial products struggle to replicate. Fermented weed tea smells extremely unpleasant. Cover the container and work downwind. Dilute at 1 part tea to 10 parts water before applying. Number 13. Comfrey liquid concentrate. Comfrey, specifically the cultivar Bocking 14, is one of the most studied dynamic accumulator plants in organic horticulture. Its taproot reaches depths of up to 6 feet, drawing up exceptional concentrations of potassium, phosphorus, and calcium. When comfrey leaves are packed into a sealed bucket without water and allowed to break down through their own moisture content, they collapse into a dark, concentrated liquid within 3 to 5 weeks. This concentrate is so strong it must be diluted at a ratio of 1 part to 15 to 20 parts water. It functions as a potassium-rich bloom booster for fruiting plants during the flowering and fruit set stage, directly supporting cell division and sugar transport in developing fruit tissue. Number 14. Grass clipping extract. Fresh lawn clippings contain 3 to 4% nitrogen by dry weight, comparable to many commercial nitrogen fertilizers. That nitrogen is bound in plant proteins and chlorophyll, which decompose rapidly when cut tissue is exposed to water and bacteria. Steep fresh green clippings in water for 3 days, keeping the container covered to reduce odor. Strain thoroughly and dilute 1 part extract to 5 parts water. Apply as a soil drench around nitrogen-hungry plants showing pale, uniform yellowing, the kind without the interveinal pattern, which typically indicates overall nitrogen deficiency rather than magnesium or iron. Do not steep for longer than 5 days, as anaerobic conditions produce compounds that can be temporarily toxic to roots. Number 15. Aged manure tea. Fresh manure is dangerous to apply directly to garden plants for 2 reasons. First, it contains raw ammonia at concentrations that cause immediate nitrogen toxicity and tissue burning, the same osmotic stress we discussed with synthetic fertilizers. Second, fresh manure from non-ruminant animals can carry pathogenic bacteria. Use only composted, fully aged manure, the kind where the pile has heated to at least 130 degrees Fahrenheit over several weeks, killing pathogens and converting ammonia to stable ammonium and nitrate. Fill a burlap sack or mesh bag with aged manure, submerge it in a barrel of water for 24 to 48 hours, then remove the bag. Use the resulting tea undiluted as a general-purpose soil drench. This delivers a broad spectrum of slow-releasing nitrogen, phosphorus, and potassium in a stable, balanced form. At this point, we have covered 15 methods, all real, all scientifically grounded, all available at zero cost. But the last 6 are the ones most people have never heard of, and the ones with the most dramatic and fastest effects on struggling plants. This is also where the most important safety information in the video applies. Number 16. Cold-processed liquid kelp. Kelp concentrates are not primarily a mineral fertilizer. They contain relatively modest levels of nitrogen, phosphorus, and potassium. Their primary value lies in natural plant hormones, specifically cytokinins and auxins, concentrated in brown seaweed tissue. Cytokinins delay cellular senescence. They slow the agging and breakdown of plant tissue under environmental stress. Auxins regulate cell elongation and root initiation. When a plant has suffered extreme heat stress, drought stress, or transplant shock, its hormonal signaling systems are disrupted. Applying a cold-processed kelp extract restores that hormonal balance faster than waiting for recovery on its own. The emphasis is on cold-processed, kelp heated during processing degrades the active hormonal compounds, leaving behind only mineral content. Number 17. Activated biochar slurry. Biochar finely ground charcoal is one of the most researched soil amendments in contemporary agricultural science. Its value is structural. Each particle is microscopically porous, with a surface area measured in hundreds of square meters per gram. In soil, biochar particles create permanent microhabitats for beneficial bacteria and mycorrhizal fungi, while also absorbing and slowly releasing nutrients and water. However, dry, unactivated biochar added directly to soil will initially pull nutrients and water away from plant roots, causing a temporary fertility crash. You must pre-charge it first. Soak raw biochar in compost tea, manure tea, or diluted worm castings for 24 hours before applying. Once saturated, it becomes a permanent nutrient retention structure in your soil that functions for decades. Number 18. Actively aerated worm casting tea. Standard worm casting tea, castings steeped in water for a few hours, is useful but limited. The most effective version involves aerating the water continuously with an aquarium air pump while the castings steep for 24 to 36 hours. Oxygenation prevents the anaerobic conditions that kill beneficial microorganisms and instead selects for and rapidly multiplies aerobic bacteria, protozoa, and beneficial nematodes. The result is a biologically active liquid containing billions of living microorganisms per milliliter. Applied to the root zone of a recovering plant, this rebuilds the suppressive microbial community that protects roots from fungal pathogen attack, the biological equivalent of an immune system for the rhizosphere. Use it immediately after preparation. The microbial population crashes rapidly once aeration stops. Number 19. Onion skin infusion. The dry outer skins of onions, the papery layers you normally discard, are a concentrated source of flavonoids, specifically quercetin and kaempferol, along with meaningful quantities of potassium and iron. Quercetin has documented antioxidant properties against several soft-bodied insect pests and suppresses fungal spore germination on contact. Pour boiling water over a large bowl of dry onion skins, allow to cool completely, strain, and apply directly to soil or as a foliar spray. This is particularly useful for indoor plants weakened by overwatering that have become vulnerable to fungus gnats or spider mite colonization. The infusion disrupts the pest environment without leaving synthetic residues on edible crops. Number 20. Blended banana peel slurry. A ripe banana peel contains approximately 35 to 50 milligrams of potassium per gram of dry weight, along with modest amounts of phosphorus, magnesium, and manganese. However, in whole, intact form, the cell walls of the peel must be broken down by soil microorganisms before any of that potassium becomes plant available, a process that takes weeks to months, depending on soil temperature and microbial activity. By blending the peel with an equal volume of water into a completely smooth liquid puree, you mechanically rupture those cell walls and expose the potassium to immediate soil solution contact. The slurry breaks down within days rather than months. 1 peel per medium-sized plant is sufficient. Apply directly around the root zone and water in. Whole or roughly chopped peels left on the soil surface invite fruit flies and rodents and block oxygen movement into the soil profile. Number 21. Human urine. This is the most nitrogen-rich, most accessible, most consistent free fertilizer available on any property, and it is the one most people will skip because of social conditioning rather than scientific reasoning. Fresh human urine from a healthy individual contains urea at approximately 20 grams per liter, along with significant concentrations of potassium and phosphorus, and is effectively sterile when fresh. Urea is rapidly converted by soil urease enzymes into ammonium and then nitrate, the same nitrogen cycle used by commercial fertilizer manufacturers. The dilution ratio is non-negotiable, 1 part urine to 10 parts clean water. Undiluted application delivers a salt load and nitrogen concentration that will desiccate and burn roots. At the correct dilution, this delivers a complete, fast-release nitrogen dose to any nitrogen-deficient plant, functioning comparably to a diluted ammoniacal nitrogen fertilizer. Apply to the soil, not the foliage, and do not apply to edible portions of plants within 2 weeks of harvest. Before we close, there is one principle that governs every single method on this list, and getting it wrong undoes all the effort. Every liquid fertilizer, natural or synthetic, is governed by salt concentration. In soil science, the term salt does not refer only to table salt. It refers to any dissolved ionic compound. Nitrogen, potassium, and calcium, in high enough concentrations, all behave as salts. They draw water out of root cells through osmosis rather than pushing nutrients in. This is root burn. It looks like synthetic fertilizer burn because it is the same mechanism. Dilution is not optional. It is the entire mechanism of delivery. A solution too concentrated does not feed the root, it dehydrates it. The rule for every liquid on this list is simple. When in doubt, dilute more. A too dilute solution feeds your plant slowly. A too concentrated solution damages or kills it. Start at half the recommended concentration and observe your plant's response over 48 hours before increasing. Yellowing that intensifies after application, wilting that worsens, or the appearance of crispy brown edges along leaf margins are all signs that concentration was too high. The second universal principle is timing. Feed your soil microbes first, with rice water, pasta water, compost tea, worm castings, and then add the nutrient source. Nutrients without active biology to process them will either leach out with the next watering or lock up in the soil chemistry. Biology is the engine. Nutrients are the fuel. Fuel without an engine does nothing. Here is the complete list one more time, with the single most important fact for each. Number 1. Green tea, cooled, unsweetened for acid-loving plants only. Number 2. Aquarium water, freshwater only, never salt water. Number 3. Coffee water, steep and strain, never apply grounds directly to soil. Number 4. Epsom salt spray, 1 tablespoon per gallon on leaf undersides, not more than every 2 weeks. Number 5. Fermented rice water, use within 3 days of fermentation starting. Number 6. Willow bark water, steep 24 to 48 hours, used during transplanting or root recovery. Number 7. Aspirin water, 1 tablet per gallon, do not increase the concentration. Number 8. Milk spray, 1 part milk to 9 parts water, morning application only. Number 9. Pasta or potato water, unsalted only, no exceptions. Number 10. Eggshell calcium, must be dissolved in vinegar until bubbling stops. then diluted 10 to 1. Number 11, wood ash tea, steep and strain, alkaline-tolerant plants only. Number 12, weed tea, 2-week ferment, dilute 10 to 1. Number 13, comfrey concentrate, 3-week ferment, dilute 15 to 1 minimum. Number 14, grass clipping extract, steep 3 days maximum, dilute 5 to 1. Number 15, manure tea, composted, fully aged manure only. Number 16, kelp, cold processed only to preserve active hormones. Number 17, biochar, pre-charge in liquid fertilizer 24 hours before applying. Number 18, worm casting tea, aerate 24 to 36 hours, apply immediately after. Number 19, onion skin infusion, steep in boiling water, cool completely before applying. Number 20, banana peel, blend with water to a smooth liquid, never apply whole. Number 21, urine, always 1 part to 10 parts water, soil application only, never undiluted. Every plant in every garden on Earth is running the same biological system. Roots absorb dissolved ions, leaves capture light, microbes mediate everything in between. The fertilizer industry exists to sell you a shortcut around the soil biology, but the biology is not optional. It is the mechanism. These 21 methods all work because they work with that system rather than around it. The plants were never asking for synthetic salts. They were asking for living soil.